Hi, my name is Brandon Vargas Koya, and this is my Group Advanced Concepts Presentation 8 for Biochemistry 1. Thank you. Using the posters that we have made throughout the past 4 weeks, I'm going to be walking through the process for how nucleotides and ribose are made, and I will show how these come together to make ATP, as well as how they are used for DNA replication. With that, I will be walking through the class, the ATP production and regeneration, ATP synthesis, as well as other information that goes along with this subject. Continuing on, we will be focusing on ATP synthase. ATP synthase is an enzyme that creates energy storage molecule, ATP, and it is the most commonly used energy currency of cells for all organisms. It is formed from adenosine diphosphate and inorganic phosphate. ATP synthase is a mitochondrial enzyme localized in the inner membrane where it catalyzes the synthesis of ATP from ADP and phosphate, as I mentioned before. It is driven by a flux of protons across a gradient generated by electron transfer from the proton chemically positive to the negative side. Focusing on ATP production, most of the ATP in cells is produced by the enzyme ATP synthase, which converts ADP and phosphate to ATP. ATP is localized in the membranes of the cellular structures, as I mentioned in mitochondria. In plant cells, for the enzyme is also found in the chloroplasts. Going along with this, how is ATP produced in the body? ATP is created through a complex enzyme-driven process. There are a couple of ways this works in cells: glycolysis, in which glucose is broken up into two subunits called pyruvate, as we had learned throughout the course of this class, which creates 2 units of ATP per molecule of glucose. This happens in the cytoplasm in both animal and plant cells. Now, focusing on the relationship between DNA and ATP, ATP is used to create DNA and RNA. Adenine is one of the bases in DNA and RNA. If you add the ribose sugar molecule to adenine, you get adenosine. If you add 3 phosphate units to adenosine, you get ATP. NTPs are used in the synthesis of RNA primers, and ATP is used as an energy source for some of the enzymes needed to initiate and sustain DNA synthesis at the replication fork. The nucleotide that is to be incorporated into the growing DNA chain is selected by base pairing with the template strand of the DNA. Ultimately, DNA replication is one of the most basic processes that occurs within a cell. Each time a cell divides, the 2 resulting daughter cells must contain exactly the same genetic information, or DNA, as the parent cell. To accomplish this, each strand of existing DNA acts as a template for replication. In molecular biology, DNA replication is a biological process of producing 2 identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as a basis for biological inheritance. Ultimately, with the aid of glycolysis and the enzyme catalyzed reactions, all of those 10 help aid in DNA replication, as well as RNA and all other forms of metabolic processes that occur in plants and animals, including animals. We're also including humans. And with that is a quick summary of the DNA replication that goes hand in hand with this process of glycolysis and ATP production. As mentioned earlier on, glycolysis does play a role in DNA replication as well as ATP production, and with that, it is a breakdown of glucose by enzymes releasing energy in pyruvic acid. Glycolysis is a metabolic pathway that converts glucose into pyruvate. The free energy released in this process is used to form the high energy molecules ATP and NADH, and as this process continues, it is a sequence of 10 enzyme catalyzed reactions. Ultimately, a summary of DNA replication involves 4 steps. Number 1, helicases unwind the parental double helix. Then, going along, number 2, the single-strand binding proteins stabilize the unwound parental DNA. And continuing on with number 3, the leading strand is synthesized continuously in the 5 to 3 direction by DNA polymerase. And with that, number 4, the lacking strand is synthesized discontinuously. Primate synthesizes a short RNA primer, which is extended by DNA polymerase to form an Okazaki fragment. Include the DNA summary for replication, along with glycolysis, ATP production, and how all these relationships form together and go hand in hand to aid in the replication and homeostasis of our bodies. Number 5, after the RNA primer is replaced by DNA, by another DNA polymerase, DNA ligase joins the Okazaki fragments to the growing strand. I hope you enjoyed my quick presentation for week 8, and I just wanted to go hand in hand and do a quick summary regarding all these different components and elements for what we have learned throughout the course of 8 weeks. Thank you.